And now I have our next speaker from the University of Colorado, Fernando Flores Guzman, who's going to talk about bone marrow dormant tumor cells that maintain reactive memory CD8 T cells. Thank you so much, Fernando, and we're happy to have you here.
Hi, good morning, good afternoon, good night. I really appreciate the scientific community worldwide is sharing their knowledge, insights, and yeah, it's great to see each one. I got to hide my face in this moment that you can see all my presentation. Let me
Yeah, it
see.
looks great. Yeah.
Good, still my face there. Okay. Thank you so much. As you know, the cancer stem cells are defined by the properties that they have self-renewal properties and they can divide by symmetry division and they give rise to asymmetry division tumor cells with different metabolic properties on one hand. On the other hand, let me then this. On the other hand, there are other tumor cells that can divide it into asymmetry and symmetry division. In both cases, you see the normally tumor cells are normally the normal tissue they can differentiate and proliferate in low rate in comparison to cancer stem cells that they are resistant to chemotherapy and radiotherapy and they are hierarchically organized like here in these red cells and this color represent different cell types within the tumor microenvironment with different immunological properties, metabolic activity, angiogenic functions. And this is the markers that you see here that I use it for this study, and other markers that can be used in the future. On the other hand, dormant tumor cells are defined the first that primary tumors are existing as normal cells, single cells individually separated, and they start to grow. They have less than this size, and they made the angiogenic switch, and they start to grow and metastasize via bloodstream or by lymphatic system, and they start to grow. This is metastasis. At some point, these cells can stay there in weeks, months, years, and maybe start to grow. And in some cases, they can start be under control of tumor immune system, specific, specifically for these tumor cells. And here you can see the markers that I use to detect dormant markers, dormancy in tumor cells, and other markers I haven't used, but you can test in the future. So the aim of this study was to investigate if dormant melanoma cells share properties and markers of cancer stem cells. We studied thereby CD133 that so far is not very well known the biological function, is present in different tumors, solid tumors, and the co-expression with CXCR4 exhibit a higher metastatic activity. CD271 is another marker studied in this study. On the other hand, hypoxia-inducible factor has exhibit a high survival motility, induced angiogenesis, and also plus CXCR4 expression is considered a master regulator of metastasis. Here you can see that normally the blood vessels are here and tumor cells that are far away from the presence of oxygen. There are in hypoxia conditions and they started to activate different genes that are characterized by stiffness and they start to grow in absence of oxygen. Under these conditions, these tumor cells can become cancer stem cells and they drive to survival, cell renewal, and tumor growth, despite of the absence of oxygen. We wanted to know if our mouse model has the presence of these cancer stem cells in dormant state. For this reason, we use a human oncogene, right? This human oncogene was transfected in a mouse model previously, and this human oncogene leads to proliferation in this CV57 mouse model and develops tumors in the skin and the lymph nodes, and we collect the primary skin tumors, we collect the metastatic lymph nodes, and we perform flow cytometry. We make single tumor cell suspension, mechanical dissociation, and enzymatic digestion. And we use a marker to characterize tumor cells using TRP2 as melanoma marker, Ki67 as a marker for proliferation. The absence of this Ki67 indicates that there is no proliferating in the cells. CD31 is a marker for endothelial cells to exclude endothelial cells. Our marker CD133 under study and to exclude leukocytes using CD45.2. So we analyze singlets, we exclude the leukocytes, the endothelial cells, and here you can see in the bottom the double staining for CD133 and the Ki67, and only very few cells are able to proliferate in CD133. The quantitative analysis, we found that primary tumor cells has a reduced amount of CD133 positive cells as compared to metastatic lymph nodes. And we found that large primary tumors and metastatic lymph nodes have a higher amount of CD133. That is reasonable. It's no surprise. You expect that when the tumor starts to progress, to grow, you have probably a high amount of CD133. And we found that these CD133 positive cells in these immunofluorescent stainings shown by CD133 in red color are localized in the peripheral areas and they are forming clusters. And the Ki67 expression is very low. Despite that this high amount of CD133 positive cells in the tumors, in large tumors, they have a reduced proliferation in advanced tumors. Now we wanted to know what is the role of these CD133 melanoma cells in our mouse model. For that, we study stem cell related markers that you can see here, 3 of the related markers, important markers, and markers related to metastasis. We found that CD133 positive cells has a high amount of these cells related to stemness and metastasis, both in primary tumors and metastatic lymph nodes. We wanted to investigate as well what is the role related to the immune response. Let me show you here first that normally the Tregs, regulatory T cells, co-express these markers, CD39 and CD73, can also express CXCR4. And these regulatory T cells usually are responsible to block CD8 T cells. They have the cytotoxic activity to kill tumor cells by secreting perforin and granzymes. But when Tregs is blocking, these CD8 T cells are unable to do this duty. On the other hand, also it's very known that antigen presenting cells can be activated by interaction, the TCR, with MHC. The second signal is costimulatory molecules that are involved as positive signals and cytokine secretion. But there are also inhibitors, molecules that are involved to block this activation, like CTL4 and PDL1. Thereby, we were interested to investigate in our mouse model if CD133 positive cells have some expression of these markers. And we found that these immunosuppression-related markers are highly expressed as well in CD133 positive cells, in primary tumors, and in metastatic lymph nodes. So we can conclude that these cells have angiogenic, stiffness, and immunosuppression functions that can drive to tumor growth. We wanted to know now what's happening in the bone marrow. We found these tumor cells disseminated in the bone marrow, as shown by these green cells. This is the marker for TRP2, melanoma-associated antigens. And animals without macroscopic skin tumors that are clinically not visible. And on the other hand, mice bearing macroscopic tumors have large clusters. In fact, we saw that these clusters, cells forming clusters, are large and in the bone marrow. However, in animals that doesn't have large primary tumors, macroscopic, without macroscopic tumors, they have the same amount of clusters. But in both groups, you can see here there are less amount of clusters sharing differentially this amount. Here you can see that macroscopic tumors have high amount of single tumor cells. And also investigating the marker CD133, there are presence of these cells in the bone marrow. And we found among TRP2 positive tumor cells, there are among 212 tumor cells and animals without macroscopic tumors, and animals with macroscopic tumors, they are for the amount 1,000 cells. This is the percentage that this is some of the concepts of cancer stem cells that only less than 1% are cancer stem cells. Also, it's important to remind that these markers for stemness, we analyze other markers. We use a CD133, the TRP2 that is melanoma-associated antigen, the markers related to proliferation that should be expressed negatively, PCNA and Ki67, and the positivity for P16, P27. There are markers also for dormant tumor cells. To go further, and we found in the bone marrow that these tumor cells are actually P67, P16, P27, and PCNA are expressed in aberrant manner. They are not colocalized in the nuclei, they are colocalized in the cell membrane and the cytoplasms. That means these really very aggressive tumor cells. On the other hand, they are in dormant state because they are not localized in the nuclei. It's also important to remember that usually the tumor cells are eliminated. The immune cells, innate immune response and adaptive immune response can eliminate these tumor cells. However, at some point, coexist the presence of tumor cells with immune cells that tolerate the presence of these tumor cells and emerge new variants of tumor cells. This is called addition that shade the presence of different subpopulation of tumor cells. And at some point, these tumor cells escape because the immune response is not anymore efficient. they escape on one hand, and the other hand, also the tumor cells manipulate the immune cells, mostly expressing these markers. So we focus on this stage, the dormant tumor cells when are coexisting in equilibrium state. For that, it's important to consider the markers that we use it as memory T cells. We use it effector memory markers for CD4 and CD8 T cells that normally they are effector memory T cells migrate to inflammate peripheral tissues in both cases, and they lack the expression of CD62L. On the other hand, the central memory T cells and CD4 and CD8 T cells, they express CD62L marker. And our research group, Umansky research group in Germany, before this study I did in Germany, in the CAF they found TRP2 specific T cells in animals with macroscopic tumors, and importantly, in animals without macroscopic primary tumors. They are still not visible. They are the existence of presence of these TRP2 specific T cells. And they found also that they have reactivity. They have reactivity also in animals that are not developing yet macroscopic tumors. And when they made adoptive transfer or isolated CD8 memory T cells from the bone marrow and co-culture with in vitro with dendritic cells, and they were injected in animals with macroscopic tumors, the adoptive transfer was succeed because could reduce or delay the tumor growth. These findings give us the mice without macroscopic tumors older than 20 weeks contain disseminated TRP2 positive melanoma cells in the bone marrow, and they can play a potential role of dormant tumor cells in the maintenance of tumor antigen specific CD8 T cells, and they are as well reactive. This is the panel we use for the stainings for analyzing the bone marrow cells plus immune cells, and we found they were colocalized and interacting in the bone marrow, as shown by these white arrows. And here you can see the interactions of these tumor cells with immune cells. Wait. Both in animals without macroscopic tumors and with macroscopic tumors, for the case of CD4 T cells and for CD8 T cells. We
So,
wanted to know if these cells are active, and we found that when there are single tumor cells, they are CD8 T cells secreting interferon gamma, they are active, but, they are not secreting perforin. However, when the tumor cells are
sorry.
large, tumors, yeah,
Yes, we are running out of time. I was wondering if you could we could jump
ah,
into the conclusions.
all right. This is the quantification of interactions, 40%. Now, we can conclude that CD133 is expressed by a fraction of melanoma cells in primary tumors and bone marrow and lymph node metastases. The majority of these CD133 positive cells are in dormant state. CD133 positive melanoma cells have an immunosuppressive metastatic angiogenic profile. They are in dormant state and they are colocalized with memory T cells. And thank you so much for your attention.
Thank you so much, Fernando. I guess we have time for only one question. It's from Sarah. She asked if all the dormant cells express the same levels of CD133, or you have like different population within the CD133 positive.
I study another potential cancer stem markers like ABCD5, CD20, CD24. I would say it's not only exclusive for CD133. We need to investigate other subpopulations. But at least in this study, yeah, they are.
Thank you so much, Dr. Fernando. There are a couple of questions in the chat. If you could help us with that,
Yeah,
if you can answer by the chat, and we think we will move on to the next presenter. Thank you so much, Fernando.
thank you so much.
Um,